Chapter 28, the reproductive system. We're going to start with the male reproductive system, working external anatomy and then working our way into the internal anatomy. So this mid-sagittal view pretty much covers everything we're going to be going over throughout this lecture. You can start at the anterior aspect, and we'll point out some of these structures now. We see that serous membrane, the peritoneum. From that peritoneum, we could work our way down into the urinary bladder, the vas deferens, all the suspensory ligaments of the penis. Remember pubic symphysis from Anatomy and Physiology 1. Some of our accessory glands, the prostate, where we will look at the deep muscles of the perineum, our bulbourethral gland, our different erectile tissues of the penis, the corpus cavernosum, and the corpus spongiosum. We went through the urethra and the three different structures and designated parts of the male urethra. We start right here with the penile urethra. We have external anatomy, our penis, the body. We have the tip of the penis called the glans. It is surrounded by foreskin known as the prepuce. And then the external urethral orifice. The root of the penis is where it is going to affix one to the body, external and internally, deep by that urogenital membrane. We have part of our scrotum and our testes, so our scrotum, our testes, and the structure that hugs the testes right here is known as the epididymis. From our movement up from the posterior aspect, we have our seminal vesicle, again, another one of our accessory structures, accessory glands. We have some bony landmarks, our coccyx, remember from that little vestigial tailbone, our rectum, our ampulla of the vas deferens. We'll talk about what that ampulla is and how it leads into this ejaculatory duct. Another accessory gland, the prostate, and the portion of the urethra that goes through it. We have the prostatic urethra here. And then we have our third intramembranous or membranous urethra, where that urogenital diaphragm goes through, so that third part of the urethra. And then ultimately our anus. This is a cadaveric look at the male reproductive system. Again, you're not going to do much with the cadaver, so you can use this just to kind of test your knowledge and see if you could identify some major structures. But again, you won't be tested on any cadavers or any pictures of cadavers as well on any quiz or lecture or lab exam. Now we're going to start with some of the external structures of the anatomy, specifically the male reproductive system. So let's look at the scrotum. We'll talk about its function, what it's comprised of, and how it serves its job. So the scrotum is a sac of loose skin and underlying subcutaneous tissue that contains the testes. Internally, it is separated by two compartments, by the dartos muscle and a subcutaneous layer. Now, let's talk about the function of the scrotum. What is its job? What is its purpose? Now, remember that the scrotum is going to serve a very important function, and that function is going to be to regulate temperature. If you think back to some of our talks, we've introduced or hinted to the fact that the job of the testes, especially when we did endocrine, is to make sperm, make these reproductive gametes. So if we've talked about the number of sperm made in a day, and we had applied a number of about 500 million or half a billion sperm that typical healthy male makes a day. That's a lot of work, a lot of energy that the male reproductive system goes and applies to this one effort. So you want to take care of them. How do you take care of them? It's about temperature, aside from nourishing. And we'll talk about the nourishing aspect, but the temperature is going to be key. Plus or minus 1 degree here can kill all that sperm. So the scrotum is going to serve a regulatory function, regulating temperature. How does it do it? Well, with two different muscles. The first is the cremaster, the second is the dartos. And these muscles work in conjunction with the skin of the scrotum. So how does it work? Well, the cremaster muscle is going to help by contracting and pulling the testes up into the body to maintain warmth. So if it's really, really cold out, this muscle will contract, the cremaster muscle will contract and pull the testes, both sides, up into the body, again maintaining a proper temperature. Now on the flip side, if it's really, really warm, the muscles will relax and allow the testes to descend and move away from the body. Now the dartos muscle serves a little different function. Its job is the same. It is going to be to regulate temperature, but it does it in a different method. How does it do it? It simply wrinkles this skin here. By wrinkling the skin, it's conserving temperatures, conserving heat while keeping the testes warm. So what happens? When it's really, really cold, it wrinkles the scrotum by decreasing surface area, ultimately decreasing heat loss. So the cremaster muscle pulls the testes up, the dartos wrinkles the scrotum to reduce surface area, to reduce heat loss. Now I like this view because it does cut the penis so that we can see our two erectile, even though these are separate bodies, that's one erectile body, and we have a second erectile body here. So we have our cavernous region and our spongy region, and we'll talk about these in a little bit. That brings us to the anatomy of the testes. So what's going on here? They are paired. They are oval-shaped glands in the scrotum, and they are partially covered by a tunic, and the tunic is called the tunica vaginalis. Now remember that this is connective tissue. It's forming a capsule, and everybody says, well, I'm confused. Why is there anything called vaginalis in the male reproductive anatomy? Well, remember, we are homolog, so we have homologous structures. They're just named a little bit differently. So if we think about the testes, we say, well, what is the testes homologous to? Well, that would be our ovaries in the female reproductive. So we both, male and female, have the same connective tissue coverings, and they are named the same thing, which is why you see some aligning with the words or the terminology. So we are, again, paired oval glands that is covered by this tunica vaginalis. It is internal, so we have another layer internal to that tunica vaginalis. It's another connective tissue layer, and it's called the tunica albuginea. Now this extends inward, and what it does is it forms these walls, these partitions. The partitions are called septa, and it's going to create compartments. And you say, well, what's in those compartments? We find seminiferous tubules. Now the job of those seminiferous tubules is to carry sperm produced within them, so they will be the site for sperm production. This is called spermatogenesis, and then it's going to help move them out of the testes into storage or into the epididymis. Now we can look at some of the anatomy and put some pictures to exactly what we talked about. So we have our testes, and we're going to start up high, and we see that we have a spermatocord. The spermatocord is going to wrap around blood vessels and nerves, as well as the vas deferens. So we see the vas deferens or ductus deferens that moves into and through the spermatocord. Now we can go to the actual compartments and the layers of the testes. So we see our first parietal layer. So this is our outermost layer. We have an inner visceral layer. Okay, so this is all parts of our tunica vaginalis. Notice these words are very similar to our serous membranes. So these are connective tissue coverings that are allowing for protection. As we move our way in, we see some seminiferous tubules that are these coiled tubules that are going to be our site for spermatogenesis or sperm production. We have a straight tubule that leads to the testes. And then we see our compartments, our septa. And these are aligned and made by our second innermost connective tissue, known as the tunica albuginea. Now, we have our septa, right? Or if you look at singular, our septum. That brings us to our epididymis, and we're going to outline this region here. Okay, that's our epididymis. And we're going to assign it regions. So the top is going to be the head of the epididymis, the middle, right here, will be the body, and the bottom portion down here would be the tail. And then we have our actual ducts. So this is where our ducts are leading to. Again, another cadaveric representation of the testes, the epididymis, and the spermatocord. Again, you're not going to be tested this way. It's just more so you can see in real life, instead of a diagram, what it looks like in the male human body. Our next step is to look at spermatogenesis. So exactly what is responsible for eliciting or starting this process of producing sperm, as well as the steps that occur. So it brings us back to a couple terms that we are going to look at today that we looked at in AP1, one being mitosis, the other being meiosis. So if you hear those words, you should be thinking, oh, it's this prophase, metaphase, anaphase, telophase. What's the difference between mitosis and meiosis? Well, sex cells versus somatic cells. We are creating haploid or 1N or half our genetic information in this process. So why that's important, especially for sex cells, is that we are combining with half from another individual, a female, to create one unique, genetically different individual. So let's work through this process. The next few slides are going to go through those steps in detail. So this picture gives us a summation, an overall view. So we see a couple of our cells, our interstitial cells, right? If you remember our Leydig cells, we hinted at them when we did endocrine, that these guys are receptive to luteinizing hormone. And this is what's really going to kick off the production of sperm. Our second group of cells are called sustentacular cells. And if you recall from endocrine that these guys are responsive to follicle stimulating hormone. So these are the pause and play for spermatogenesis. So interstitial cells are going to make testosterone. Testosterone is going to increase the ability for these sustentacular cells to work and these seminiferous tubules to make more and more sperm. So stands to reason that if you have low T or low testosterone, your ability to make a decent amount of sperm is diminished, which is why as males get older, especially over the age of 30, their testosterone levels drop, so does the number of sperm they make a day. Now, let's look at some of our structures. We have some blood capillaries that is nourishing this area. We have a basement membrane. Now, below that, we have our process, our spermatogenic cells. So these stem cells, they're called spermatogonium. Now notice that these stem cells are diploid. They are 2N, so they contain all the male's genetic information. We undergo our first process of mitosis, bringing them down into a primary spermatocyte. And again, we're going to look at this in detail, so don't get too crazy or worried about what we're talking about. But I want to point this out now. We are still diploid. And then something happens, another process of meiosis, and then we undergo a split into 1N. This gives us a haploid state. It is called the secondary spermatocyte, at which point we're going to enter a different process. The process above is called spermatogenesis. We are now into spermiogenesis. And I'm going to write the word down so you can see it, spermiogenesis. It's a little bit different of a process. It is the physical maturation of this sperm. Now we enter into this spermatid. Again, it is haploid, at which point it will mature and differentiate into a spermatozoa, or mature sperm cell. So we can name that terminology. So we said that the primary spermatocyte undergoes meiosis 1 to become 2 secondary spermatocytes. Here's our first haploid cell. Meiosis 2 takes place and the secondary spermatocyte becomes 4 spermatids. Now this is important. If we're making a 500 or half a billion sperm a day, that we take advantage of all these cell divisions. So we start with one stem cell that is going to give rise to ultimately 4 spermatids. Now we can put this entire process of spermatogenesis to picture. So we see where mitosis occurs and where meiosis 1 and meiosis 2 occurs, and why this is unique to us humans, well, to all animals, but why it's super important, especially in this one stage of meiosis. We'll talk about why and what's going on here. But let's look at our first start. We start 1 with our spermatogonia, that's our stem cells. Again, we're 2N. We undergo mitosis, prophase, metaphase, anaphase, telophase, and we know that our end result is still going to be a 2N cell. Now, with differentiation, our primary spermatocyte is going to give rise to our secondary spermatocyte. What happens here? We are replicating our DNA, we're forming these tetrads, and more importantly, we are crossing over. So, what is crossing over? This is going to be super important for human anatomy. So we say, what exactly is crossing over? So it occurs at a specific point between prophase 1 and metaphase 1, and it's where 2 homologous chromosomes, non-sister chromatids, pair up and exchange various random segments of genetic information. And basically they're forming 2 recombinant chromosomes of sister chromatids. Now, what does this mean? Because this gets genetically complex. Well, let's make it really simple. So what this does is it ensures diversity of our species. It makes sure that not one sperm will ever be genetically the same as another. Now that's super important. You say, well, why is that important? Well, think about you or your siblings or your family, and if you have a non-monozygotic twin or non-identical twin, your brothers and sisters are completely genetically different from you. And the reason being is because of this crossing over. If you think about the male sperm making 500 million sperm a day, how do you make sure that you are truly a unique individual? Well, that's crossing over. Your parents will never, ever, ever, and you could run the numbers, make another sperm and another egg and get it to recombine exactly the same way to make another you. And that's important for the human species. We don't want to shorten or reduce our genetic diversity by not having this event. So we talk about founder effect and bottleneck effect, and I usually use an analogy in my class of a bottleneck effect scenario combined with the founder's effect scenario to explain why crossing over is so important. So if you think about our classroom, we take 80 kids, say we had an even number of boys and girls, we go on a boat trip, and the boat gets crashed and stranded on this deserted island that nobody knows about. Our genetic diversity upon the crash is pretty high. We have 80 unique individuals. Well, over time, those individuals are going to mate, and they're going to make a first generation. Now, what happened to the gene pool? It was reduced. Now, that first generation is going to mate over time again and make a second generation. Now, our gene pool got even smaller. So what's happening? You can envision that upon arrival, our genetic diversity was so big. As they start mating, that genetic diversity starts coming closer and closer. This is known as the bottleneck effect. And what happens is it gets to the point where we're going to get into our fifth and sixth generation, so F6 is going to start inbreeding. And what happens to populations that start inbreeding? They usually become extinct. So nature and evolution had to ensure that crossing over, or there was a process such as crossing over to ensure that this bottleneck effect did not occur. So how does it do it? It makes sure that every sperm, every egg created by the male and female body is genetically unique. So hence crossing over. So a little digression, but to explain what crossing over and why it's so important. So after that first breakup for meiosis 1, we enter into our secondary spermatocyte. Now each chromosome has 2 chromatids. We enter into meiosis 2. So we're breaking down again through prophase, metaphase, anaphase, telophase 2, and we end up with our spermatids. Now, look at our terminology. The all-encompassing process is called spermatogenesis, and where we're allowing for the physical maturation of sperm into what we typically think sperm looks like is the process of spermatogenesis. And again, this is occurring internally. So we differentiate and we start forming some of our structures that are typical with a mature sperm. And we'll talk about what these structures are in a little bit. What is the function of sperm? So sperm is designed to reach and penetrate the secondary oocyte. So we're talking about the egg in the female anatomy. And it needs to do this in order to achieve fertilization and create a zygote. The head of that sperm contains a nucleus that has about 23 chromosomes, right? So that's important. Remember, we are dealing with half or 1/n of our genetic information. We have an acrosome that covers the head, and this is going to contain enzymes. These enzymes are going to be released cooperatively with the other sperm. If you envision throwing a massive amount of bread into Times Square or into a park, and all these pigeons come flying down attacking all that bread, think about sperm acting like that. And every time they peck and hit into that egg, they're releasing these enzymes that are going to slowly penetrate into that oocyte. The neck contains centrioles that's going to form the microtubules that make up the rest of the tail. The mid piece, or the middle piece, is going to contain mitochondria. That's going to make ATP for locomotion of the sperm. Now remember, sperm is a flagellated structure. It's the only flagellated structure in the entire human anatomy, and that's unique. The principal piece and the end piece make up the tail that's going to whip around and allow the sperm to move and become motile. Now let's look at what all these structures are. So again, we have the enzyme-filled acrosome, so this is the top part of our head. We have our nucleus with our half 23 chromosomes, so half our genetic information, our neck, our midpiece that contains the mitochondria to produce energy. But now the next question is, where do we get the energy to feed the mitochondria so that we are producing enough energy to run the tail? Well, that's going to come in the form of sugar, and we'll see where this sugar comes from in a little bit. So here's our principal piece and our end piece that's going to whip around, allowing this to kind of swirl like this, swimming it in a forward direction. Now, we introduced and talked about some of the hormones that control spermatogenesis and control testicular function. So let's reintroduce them. And put a lineage, a time frame into what happens. So at puberty, gonadotropin releasing hormones, so GnRH. So this is from, you should be thinking, where's all these releasing hormones from? So the hypothalamus, right? So the hypothalamus is releasing gonadotropic releasing hormone. It's going to stimulate the cells in the anterior pituitary. Now, what type of cells? We have gonadotrophs. So remember those cells that we discussed in endocrine that made our sex hormones? Well, here we are putting it full circle. So our gonadotrophs are being stimulated to produce and secrete luteinizing hormone and follicle stimulating hormone. Now, in our previous slide, we said what they do, but let's reaffirm it. So luteinizing hormone stimulates cells in the testes, and we say, well, which cells? So they're Leydig cells, right? So Leydig cells are the one we're talking about. So luteinizing hormone stimulates Leydig cells to produce testosterone. The follicle-stimulating hormone is going to stimulate spermatogenesis by stimulating those Sertoli cells, so S-E-R. Sertoli cells, also known as sustentacular cells. So this is what's really going to kick off the process of spermatogenesis. Now, FSH is kind of like the pause and play button. This will signal the creation of sperm or pause it when that male made enough sperm for the day. Or say after an ejaculation, we are at a deficit. Here comes FSH to signal spermatogenesis, talking to those Sertoli cells to make more sperm. So let's look at our feedback loop. So we start with gonadotropic releasing hormone. It moves its way to those gonadotrophs. There's those gonadotrophs secreting FSH and LH. They're going to move through the blood, and it's going to attach to FSH receptors on those Sertoli cells, right? Now, once it attaches to those FSH receptors, spermatogenic cells become active. The sustentacular cells are going to secrete androgen binding protein, which is called ABP, which is going to stimulate sperm production. Now, we look at the negative aspect of feedback. Here comes inhibin. Once we made enough sperm, inhibin is going to be secreted, which is going to go back to the anterior pituitary and decrease release of FSH. Again, here's our pause and play scenario. Now, together with testosterone, FSH stimulates spermatogenesis. Now, where does that testosterone come from? Well, that's where LH comes in. LH passes through the blood. It's going to stimulate testosterone by binding to those LH receptors on our interstitial cells or Leydig cells. It's going to produce testosterone. Now, before we get into our negative feedback of this, what's testosterone do? Now, we have another intermediate structure or function going on here. So here's our testosterone. Testosterone is going to work on Sertoli cells to maximize sperm production, as well as lead to enlargement of male sex organs and expression of male secondary sex characteristics. This starts at puberty. So we talk about the deepening of the voice, the enlargement of the Adam's apple, the change in tension and strength of those vocal folds. We talk about hair growth under the arms, around the genital area, face, chest, back, etc. These are all secondary male sex characteristics. We also deal with another process that we didn't do with digestion, which was metabolism, and a specific subset called anabolism. And anabolism is protein synthesis. Now, let's go to our negating factor. Here comes testosterone. It's going to talk to those cells to decrease LH production, as well as release of gonadotropic releasing hormone. So again, shutting this system down. Our next few slides are summary slides. And again, I usually highlight words in red that are important to kind of point out some of the key terminology. So we start with spermatogenesis. It begins in the seminiferous tubules, gives rise to these spermatogonia. These are stem cells involved in this process. The spermatogonia are going to lead to our primary and secondary spermatocytes that lead to, we should have an arrow here, that leads to our spermatids, then to our spermatozoa. We have our sustentacular cells. We also call them Sertoli cells. This is going to sustain and promote development of spermatozoa. And then our main hormone again, FSH, that's going to help stimulate this process. That gives us spermiogenesis, that last phase of spermatogenesis. And we talked about those specialized cells. The sustentacular cells are important during this phase, the pause and play scenario, that FSH that's stimulating it, and then the inhibitory inhibin. So again, here's a nice hormone that is telling us that it is inhibitory. Right? So inhibin sounds like inhibitory. It's exactly what its function is. And that sustentacular cell maintains the blood-testes barrier tight junctions. And if you look at a previous slide, you'll see where exactly that is. As far as function, the blood-testes barrier is one of the tightest blood tissue barrier in the human body. It's going to divide the seminiferous epithelium into two layers, a basal and an apical, which is also known as an adluminal compartment. Basically, meiosis 1 and meiosis 2, spermiogenesis and spermiation all take place in a specialized compartment, a microenvironment behind the blood-testes barrier. Now the question is, why is this important? So this is necessary to avoid the production of anti-sperm antibodies and prohibits autoimmune diseases which can lead to male infertility. So as you can see, this is a super important barrier, and it's creating this immunological barrier as well as a physical partitional barrier. Before we start getting into detail some of our other structures, let's look at our summation of the male reproductive system. So some of our hormones, testosterone and dihydrotestosterone, produce several effects that we have talked about before. One, that prenatal development, so the enlargement of the male or signaling in utero of those male sex organs that are the penis and the testes. We deal with development of those male sexual characteristics, development of sexual function with testosterone, and stimulation of anabolism. Now, we really haven't talked about anabolism, again, because we didn't do metabolism after digestion, but just to give you a quick understanding of what anabolism is, it's a basic process where these smaller, more simpler molecules are pretty much combined into larger complex substances. So our body has this unique ability of being able to assemble. And what it does is it utilizes energy. And you say, well, where does that energy come from? Well, it comes from catabolism. So anabolism is fed by catabolism. And basically, catabolism is the breakdown of larger, more complex substances into smaller ones. So it's kind of the opposite of anabolism. So it utilizes energy, the complex chemicals it needs by combining these smaller molecules that are derived from the foods we eat. So catabolism is breaking down from larger to smaller, where anabolism is making bigger from smaller to bigger. And then we have our negative feedback system that controls the blood level of testosterone. That leads us to that feedback loop. So we have a stimulus, disruption is going to increase blood levels of testosterone. The cells in the hypothalamus that secrete GnRH are going to be signaled to be decreased in portal blood. The anterior pituitary cells, those gonadotrophs, are going to be signaled to decrease LH in systemic blood. The effect are those interstitial cells are not going to be signaled anymore because LH is not binding to those LH receptors. So we secrete less testosterone. The response is decrease in blood levels of testosterone. We return to homeostasis. So again, our feedback loop. Now that we spent some significant time talking about sperm creation and the site thereof, let's talk about how we move that sperm out of the penis for reproduction. So we have this system of ducts in the male reproductive system. Sperm and fluid travel from the seminiferous tubules to the straight tubules and then to a network of ducts, the retestes. These efferent ducts are going to carry the sperm to the epididymis. Sperm is going to mature in the epididymis, and we're also going to degenerate sperm here and are going to reabsorb them. So what's the function of the epididymis? It's going to allow for the maturation of sperm, and it's also a recycling plant. If that sperm is not ejaculated, there is a lifespan for sperm. It will be degenerated, it will be reabsorbed. If sperm is detected to be dysfunctional, some of these will be destroyed and reabsorbed as well. The epididymis will then propel sperm into the ductus deferens or the vas deferens. So let's look at that epididymis. We already said that there is a head, a tail. So here's our head, our tail, and our body. So the epididymis begins at the superior testes and runs along the posterior border before connecting with the vas deferens. So we see our vas deferens moving this way, right? So that's where it's connecting at the tail. We see three main functions: monitoring and adjusting composition of seminal fluid. It's recycling damaged spermatozoa and cellular debris by enzymatic breakdown. And it's storing spermatozoa, facilitating their maturation into functional sperm. This maturation process completes here, although sperm are still not motile yet because they do not have enough fluid to be able to swim. So we rely on smooth muscle contractions within these tubules to propel and move sperm to our first set of accessory glands that is going to produce the majority of the fluid so that sperm can become motile. Let's look at some of this internal anatomy. So what we see looping over the bladder is our right and left, okay? So our ductus deferens, right, or vas deferens. Now, we're looking from the posterior view. That's why our right is on our right and our left is on our left. So we're looking from behind so we can see all these structures. Now, we have our seminal vesicles, our accessory gland. We'll talk about this in a little bit. We have our bulbourethral gland, another accessory gland, and a prostate. Now, a couple things to point out. The vas deferens leads into an ampulla, a storage chamber, and then our first accessory gland, our seminal vesicle, is going to merge, forming this ejaculatory duct. And this ejaculatory duct is going to do just that, push sperm into the urethra so that it can be expelled from the penis. A couple other things to point out. We have the bulb of the penis here. Here's our urogenital diaphragm right here where we find our intramembranous or membranous urethra. We have the crus, this forking of the penis. And from a lot of our sagittal cuts, we're not able to see this. This is known as the crus of the penis or root. Its job is to give these erectile tissues strength and stability. That's why it is known as the root, to kind of like the root of a tree that gives a tree stability. This, when erect, will keep that give that penis some strength and stability as well. So our vas deferens exits the tail of the epididymis and ascends through the spermatic cord into the pelvis. It loops over the ureter and passes over the side and down the posterior surface of the urinary bladder, as we just saw on the slide. This is going to give us a cadaveric view. So again, you won't be tested on this, but kind of a game to see if you can identify some of these structures in a real live human model. The spermatic cord is going to ascend out of the scrotum and contains the ductus deferens, testicular artery, veins draining the testes, autonomic nerves, lymphatic vessels, and the cremaster muscle. The spermatic cord and the ilioinguinal nerves pass through the inguinal canal, which originates at the deep inguinal ring and ends at the superficial inguinal ring. So we can put all that terminology to structure. So we have our spermatic cord up here. We have our superficial or cutaneous inguinal ring. You can see that cremaster muscle, ductus deferens or vas deferens, our autonomic nerves, testicular artery, green lymphatic vessels, our pampiniform plexus. These are forming our testicular veins, our epididymis, all our tunicas, allogenea, vaginalis, our internal spermatic fascia. We have our raphe. This is that little crease. which marks the uniting of the two halves during embryological formation. We have the skin of the scrotum. There's our wrinkling dartos muscle, our external spermatic fascia, cremaster muscle again, scrotal septum, our penile or spongy urethra. We have our corpora cavernosum on top, our erectile tissue, our corpora spongiosum on the bottom, that's containing the penile urethra. We have some suspensory ligaments of the penis, as well as the fundiform ligament of the penis, and our aponeurosis. So it's our external cut of the oblique muscle. So that is the connective sheathing that connects those muscles. So again, a lot of these words you don't need to know. I'm just pointing out every single structure on these models. That brings us to the ejaculatory duct. The ejaculatory duct arises from the junction of the duct from the seminal vesicle and the ampulla of the ductus deferens. At this point, the urethra is the duct shared by the reproductive and urinary system. So both semen and urine pass through the urethra. And we're going to see how the accessory glands are going to account for this shared tube. Remember, urine tends to be very, very acidic, and semen tends to be very, very basic. So we have to figure out how to deal with this issue. So the urethra passes through the prostatic glands, this is a prostatic urethra, deep muscles of the perineum through the intermediate or membranous urethra, and then the penis, which is called the spongy urethra. Again, let's put this to picture. We see our bladder all the way on top, our prostate with our prostatic urethra. We go through the membrane right here for our membranous urethra, through the penis for the penile urethra, and out the external urethral orifice. Now we can talk about the meat and potatoes of the system, the accessory glands, which include the seminal vesicles, the prostate, and the bulbourethral gland, also known as Cowper's glands. We're going to start with the seminal vesicle. This is the first gland that's going to contribute the majority of the volume of semen. So we have an active secretory gland. It contributes about 60%. Now, what does it contribute? It secretes fructose, sugar. So remember, we alluded before that sugar was necessary to produce or supply nutrients for spermatozoa. Think of that sperm as a motorboat. What does a motorboat need in order to run the engine? Well, it needs water and gasoline. So the water, so if you think of our sperm as being that boat, the water is this volume of semen, this fluid that we're contributing. So at this point, sperm is really able to move their flagella and become motile, right? So once they're exposed to these secretions, they can actually swim now. So that sugar is essentially the gasoline. So now at this point, sperm is here. We have the water so it could swim, and the gasoline, the sugar, so that we can provide nutrients for that engine. Then next is prostaglandin. So look at the word, prostate. So this is going to stimulate smooth muscle contractions along the male reproductive system, specifically at the prostate. Then next is fibrinogen. This is a plasma protein. So we say, well, this is interesting. Why is this seminal gland, the seminal vesicle, producing a protein? And if we go back to blood, we remember that this is for clotting. So why fibrinogen? Why clotting? Now, in order to understand this mechanism, we have to think about where the sperm is going and what's the purpose of all these secretions. So as we move through these accessory glands, remember that they're supplying nutrients for sperm. They're contributing an amount of fluid, okay? So what's this fluid doing? Well, it's allowing the sperm to be motile. It's providing nutrients for the sperm, but there's a more encompassing goal. We are going, and if you think about where this is going into the female vagina. So think about what's happening here. The female vagina is a very acidic, dangerous, hostile environment for sperm. So we have to find a way to protect it. The fibrinogen essentially is a shield. So if you think about sperm now being in a female body, and what does the female body say? Invader. It sounds the alarm, right? So what comes? White blood cells. These white blood cells are going to come and attack and try to kill the sperm. Now, if we allowed the sperm to be killed by the immune system of the female body, we wouldn't be very successful at reproduction. So we came up with a protective mechanism, hence fibrinogen. Fibrinogen is going to clot these cells in the female vagina, essentially blocking, shielding the immune response to the invasion of sperm into the vaginal canal. The last thing we're going to see here, the seminal vesicles produce this alkaline pH. So what's alkaline mean? We're talking basic, right? And if you've ever touched bleach with your hands and you see how slimy it feels, it's because bleach is very, very basic. Well, the texture and that slimy feeling of semen is because of this high basicity. So again, why? Why are we making it so basic? We are neutralizing. Remember, we just said that the vagina is very, very acidic, so we have to remove that acidity by, again, protecting the sperm by making it basic. So again, at this point, spermatozoa sperm begin to move their tails, their flagella, and they are motile, and again, they're good to start swimming. The next accessory gland is the prostate. So what does the prostate do? It contributes about 30% of that seminal fluid. Now it's going to secrete a slightly acidic fluid. So we say why? This is where we start getting into pre and post ejaculatory secretions. This is going to be our pre-ejaculation. What's happening? We are cleaning the urethra. So we secrete this acidic fluid to cleanse the urethra of any casts, microbes, or any urine that remains. Remember, this is a shared tube. The second secretion is seminal plasmin. This is unique to males. Females do not have this. This is a natural antibiotic. And basically what this does is it cleanses and protects the urethra from growing bacteria. This is why males have a lesser incidence of getting UTIs, urinary tract infections. The last gland is those small bulbourethral glands lying next to the bulb and the urethra of the penis by that urogenital diaphragm. It secretes 5% of seminal fluid, and it's going to be mostly alkaline mucus with these lubricating properties. So again, we're neutralizing the secretions within the vagina while lubricating the tip of the penis to facilitate movement into moving the penis into the vagina. Now let's talk about the overall composition of semen. Now remember, the average ejaculation's about 2.5 to 5 mils of stuff. Within this stuff that we just said, so this 2.5 to 5 mils in that ejaculation, we have our fluids from our glands, as well as anywhere from 50 to 150 million sperm per milliliter. So if we take the average of that and we say, well, let's call it 100, and then we take on the high end of the ejaculation about 5 mils. So for every ejaculation and for every 5 mils of stuff that comes out, we're dealing with about 500 million sperm. So that's a lot of sperm per ejaculation. 500 million sperm per ejaculation on average. Again, for an average healthy male, 5 mils of stuff. Now look at this. What percentage of sperm makes up that ejaculation? So we have 500 million sperm and about 5 mils of stuff. You're probably running through some numbers. I'd probably ask you to guess, but since this isn't interactive, I'm going to tell you. It makes up less than 1%. That's crazy, right? Less than 1% of that ejaculation is actually sperm. That means that 99% of what comes out of the penis is there for nourishing the sperm, and more important, we're going to put it in big words, protection. We are protecting the sperm from the hostile environment of the vagina. The pH is going to be about 7.2 to 7.7. This again going to be more on the basic side to help neutralize the vagina. The penis contains a urethra, and it is that passageway for semen and urine. We have 3 cylindrical masses that we've alluded to but haven't outlined yet. We will. 2 corpora cavernosum, 1 corpus spongiosum. We have the glans or tip or head of the penis covered by foreskin known as prepuce. Now, that prepuce is usually removed depending on your ethnicity, religion, or location, basically where you live. This is known as a circumcision. So let's look at some conditions that can arise due to disruptions in homeostasis. So remember that prostate. It is about the size of a chestnut. It is not a paired organ. The
seminal gland and the bulbourethral glands are paired. The prostate is not. It wraps around the urethra. So what happens? We can see a condition known as benign hypertrophy of the prostate. Basically, as males get older, the levels of testosterone decrease. The prostate will grow, and it can obstruct the urethra and cause possible kidney damage. So how big can this thing get? It could go from the size of a chestnut, especially with malignant growth. It can get to the size of a grapefruit in a small area. Now, what happens with benign hypertrophy? So they usually do a procedure called a transurethral prostatectomy, and this is called a TURP. And basically they insert an instrument into the urethra and they kind of roto-rooter or cut away excess tissue, opening up that urethra again. The second condition is actual prostate cancer. While it is prevalent in American males, it is not the most deadliest cancer. Believe it or not, testicular cancer is one of the most aggressive and deadliest cancer for males, as is ovarian cancer for females. Now, this is malignant growth, usually detected during a rectal exam. There's also blood tests that you can use to check for prostate cancer. It's known as PSA, or prostate-specific antigen in the blood. So basically, men over the age of 50 get an ultrasound that can measure the size of the prostate. You can also do it digitally with rectal exams. This can be treated with radiation or surgery before it metastasizes, but if left untreated, the prognosis is not very good because this cancer will then be spreading, hence the word metastasizing, to different parts of the body. Which brings us to testicular cancer. Now, for the types, men in their 30s and 40s, this can grow quite rapidly. Symptoms: we get discomfort, pain, or heaviness in the scrotum, changes in testicular size, shape, or texture. Here's the diagnosis. Self-exam. Run your finger along the margin of your testes. If it is not smooth and continuous and you have these larger hardened bumps, this can indicate some abnormal growth. CAT scans, blood tests, tumor markers, CXR is other ways that we can monitor or check. Look at the x-ray to the right. All these tumors in the lungs. Testicular cancer spreads quite quickly to the lungs, right, which can lead us to stage 3 cancer. Treatment depends on the stage. Right? If left to grow quickly and unimpeded, the prognosis is not that great. Or if caught early enough, we can use surgery or orchiectomy, which is to remove the testes, chemotherapy, or radiation. So we've talked about the accessory glands, we've talked about sperm, we haven't really talked about the penis. How does the penis deliver sperm into the vagina? It needs to become erect, rigid. So it does that with 2 erectile tissues, one called the corpora cavernosa and the other called the corpus spongiosum. So look at the corpus spongiosum. It is 1 erectile body, and the corpora cavernosa is 2. So again, why these names, caverns and sponges? Well, they're named after their job. This penis becomes erect with engorging or filling of blood. So caverns and sponges are great at holding blood, so it's a proper name for these tissues. Now notice the deep arteries, so highly vascular, that's going to deliver blood. We also see above it on the dorsal aspect, we see a deep dorsal vein, a dorsal artery. We have some fascia, a subcutaneous layer, and a superficial dorsal vein. So we see that again, a superficial dorsal vein, a deep dorsal vein, again, taking blood to and from the penis. So again, we need this to engorge the penis to become erect. On our next slide, we'll look at some of the processes. So how do we get an erection? It is brought about by parasympathetic innervation, leading to vasodilation of arterioles in erectile tissue. Now notice I said parasympathetic. Now remember fight or flight. This is problematic for males. So we say, why is it problematic? So with fight or flight, we're diverting blood away from the parasympathetic system and putting it to our muscles to sustain fight or flight. Now, what causes fight or flight? Well, we could think about stress as an example. So when a male is stressed, his fight or flight system is activated and the parasympathetic is suppressed. So what does this mean? So if a male is under undue stress, he will have a hard time maintaining an erection. Okay? So we can see some complications. So large amounts of blood enter the tissue into those dilated blood sinuses. Ejaculation is the powerful release of semen due to sympathetic stimulation. So again, this is a positive feedback loop that's brought on from our sympathetic stimulation. And then we have the bulbospongiosis, the ischiocavernosus, and the superficial transverse perineal muscles that are going to produce these strong contractions that are going to force the semen out. So we could draw a big arrow, force the semen out of the penis into the vagina. Now we can review some hormones. So what hormones work with the male reproductive system? So again, repeat in red, we see some of our basic hormones with their function. FSH, as we talked about, targets those sustentacular cells to promote sperm production. Luteinizing hormone causes secretion of testosterone from those interstitial Leydig cells of the seminiferous tubules. Here's our gonadotropin releasing hormone released by the hypothalamus that targets the gonadotrophs in the anterior pituitary to release FSH and LH. And lastly, testosterone that's released by the hypothalamus to target the pituitary gland to release that FSH and LH. So, as we transition away from male into female, I usually like to bring up this chart, and it didn't translate very well from our slide to explain everything, but let's look at some of the differences between males and females when we talk about production of gametes, when that begins, when it ends, where and how it matures. So let's look at this table. So we talk about male versus female. Now, we've outlined the male system, but we haven't talked about the female. So let's go back. We know that for every cell, males are going to make 4 gametes, okay? So 4 sperm. The production begins at puberty. So males aren't making sperm until they hit puberty. And we know that because this is going to signal the secretion of testosterone, which kicks off the system. Now, the next question is, when does production end? Well, males can produce sperm all the way to the ripe old age of, believe it or not, 80. So males are making sperm, although they're not making as many sperm, that 500 million a day that you get in your early teens or late teens and early 20s and maybe 30s, but they are ultimately making sperm to the age of 80. Now we look at the maturation. Now this is really where that slide got messed up in that translation. So the maturation is going to be dependent on secretion from the glands. So dependent on secretions from the glands, okay? So those accessory glands is also going to allow for the maturation of that sperm. Now this is where females are really different. Let's look at our number of gametes. It's typically 1. Why do we say usually? Well, sometimes we could get 2 eggs expulsed in one reproductive effort. This can lead to twins, dizygotic twins. So why? Let's talk males versus females. Males are an on-demand system. We need more sperm. We make it, right? Females are going to be a little bit different. Look at their production. It begins in utero. So you are born with all the eggs that you will have through your entire lifetime. You are not an on-demand system. You are a finite system. So you have a finite number of eggs. When does production end? Well, once all those eggs are gone, you enter into menopause. Now, maturation. So this is where our slide got a little messed up, but it's going to sustain itself with the corpus luteum. And when we get into female reproduction, don't worry about the terminology, we will get into it. So the corpus luteum is going to sustain it until the placenta takes over. Okay? So again, basic differences from males to female. 4 gametes to 1. Production begins at puberty. Males in utero. Females, production in old age up until 80 for males. And menopause for females. That age can vary. Right? It depends on when that female hits puberty. And again, we'll talk about this when we get into female reproductive system, which is on our next slide. The female reproductive system. So we're going to discuss the female reproductive system in a similar fashion that we did the male. We're going to start with external anatomy and work our way to the internal anatomy, structure, and function. So we start with the external anatomy, and this mid-sagittal cut takes us through some external structures. The mons pubis, being this fatty outer vaginal pouch, the clitoris, the labias majorum or majus, minor, major, labia minus or minorum. We have our openings, our external urethra orifice, our vaginal orifice or canal. We have our anus in the posterior aspect over here. So that's pretty much our external anatomy. As we get internal, we'll look at some of these structures. Again, another true human cadaveric view of the female reproductive system. See if you can find some major structures like the uterus, the mons pubis, the clitoris, the two labias, major and minor, the vagina, the rectum, urethra, etc. So let's look at some of our structures. So we started with the testes in males. We now are at the ovaries. Now, let's talk about what they are. They are paired glands. They are homologous to the testes. Size and shape. They're about the size of an almond. Yes, an almond. So next time you have an almond in your hand, hold it up, look to your friend to the right and say, these are my ovaries. Their job is to produce gametes. These are going to mature into your ova. And we have hormones that are responsible for kicking this process off, such as progesterone, estrogen, inhibin, and relaxin. The ovaries are supported by a ligament called the broad ligament, an ovarian ligament, and a suspensory ligament. Let's take a transverse cut and look down onto it. So what can we see? We can see the ovaries, those two white structures there. We see a ovarian ligament tying it into the uterus. Okay, so we're dealing with the uterus right here. We have a round ligament. So this round ligament is just that. It is holding assisting to hold the ovary in place. We have a mesovarium, so it is this little ligament that goes above the ovarian ligament binding the ovary to the fallopian tube. We have a broad ligament, and we need a different view to really see how broad this ligament is. We have a uterosacral ligament, so again, tying that uterus into the sacrum. We have a suspensory ligament, so suspending these structures in place, the fallopian tubes in place. Remember, key is holding this system in place. You don't want your fallopian tubes, your ovaries bouncing and hitting against the uterus every time you jump or move or walk. Now we can look at the histology of these structures. Histologically, the ovaries are covered by a germinal epithelium. Then we see some consistency. Look at our capsule and its connective tissue. We see this again repeating in the male, the tunica albuginea. It is a capsule of dense irregular connective tissue that is located below that germinal epithelium. We have an ovarian cortex. It's below the tunica albuginea and consists of ovarian follicles and stromal cells. That brings us to the ovarian medulla. So we're now in the middle portion of this structure where you find connective tissue, blood vessels to bring those hormones and nourish these cells and structures, lymphatic vessels, and nerves. Focusing in on those ovarian follicles. What's in those follicles? Now look at our hormones. So we start bringing and coming full circle. Follicle stimulating hormones. So this is the follicle we're talking about. Within those ovarian follicles are going to be our oocytes. They're going to be in various stages of development. Follicular cells and granulosa cells are going to be located in here. We'll talk about what those are in a little bit. Entering into our tertiary or final stage of maturation of this follicle, it's called the Graafian follicle. This is when it is enlarged and closest to the surface of the ovary, where it's ready to rupture and expel that secondary oocyte, that egg, into the fallopian tubes. The corpus luteum is what develops after ovulation, and this will persist as an endocrine structure. Once the egg is emptied, the follicle will produce hormones, progesterone, estrogen, inhibin, and relaxin. Depending on whether the egg gets fertilized, will decide what happens to the structure after ovulation. let's take a look at the ovary. Now we do have a tutorial on Anatomy and Physiology Crack that goes through a model for lab, so this will tie directly into lab. So we're going to come full circle. We're going to start at the primordial follicle. This follicular nest is what you're born with. So we're born with, on average, about 300,000 eggs. By the time you hit puberty, you lose about 2/3 of that. You get about 100,000 remaining. This is what you have to ovulate from puberty all the way through menopause. Okay, so that primordial follicle will differentiate into a primary follicle, and then that follicle will grow into a secondary follicle, and we see that antrum is starting to form. This is a fluid-filled cavity. All right, and we start seeing that ovum being surrounded by these granulosal cells and a layer of cells called the corona radiata. And then at our largest, our tertiary follicle or our Graafian follicle, there's that fluid-filled antrum. Okay, and then we have ovulation. The egg is expulsed, the fluid ruptures, and the corona radiata, so radiating crown, is going to protect this layer of granulosal cells, are going to protect the discharged secondary oocyte or ovum. This is ovulation. Now, this follicle will degenerate into something called a corpus luteum. It will pulse hormones, specifically and primarily progesterone. Progesterone is our communicating hormone. It tells the body if we're pregnant or not, if we fertilize that egg or not. Now, if that egg was not fertilized, hormones will cease to be pumped from this structure. It degenerates into something called the corpus albicans. The corpus albicans will persist as scar tissue along the margins of the ovary. Now, how do we form these eggs? The formation of the gametes in the ovaries is called oogenesis. Remember, in males it was spermatogenesis. Here it's oogenesis for oocyte. It begins before a female is born with the process of meiosis, at which point it's paused, and it will wait until that girl hits puberty. When the primordial germ cells migrate from the yolk sac to the ovaries during fetal development, they differentiate into oogonia. The oogonia divide into germ cells, some of which become primary oocytes. Let's put it to picture to understand what's going on here. So our overall process is oogenesis. So before birth, we undergo mitosis and we're stuck in this primary oocyte. Notice it is still diploid. From here, we are paused. So this is those roughly 300,000 eggs that were created that are sitting in those follicular primordial nests. And it's paused here after mitosis. Now, before birth, we are undergoing DNA replication. So we're forming our tetrads. And again, we're still paused. Then, at puberty, we undergo meiosis 1. Now remember, meiosis 1 is where we're going to have our crossing over, but now we have an issue. Remember our previous table. Males make 4 gametes, females make 1. When we undergo meiosis, we should expect to see a second cell propagating, under which that cell will undergo meiosis 2, and then we end up with 4, but we don't see this. Why? That second cellular product is called our first polar body. It will get kicked out, and it may or may not undergo a second stage of meiosis, but either way, it will be degraded and digested and removed, allowing for the best product to persist. Now, this is called our secondary oocyte. Realize that we have now become haploid. So our tetrad formation formed at our primary oocyte, we undergone meiosis, we have our crossing over, and gives rise to a haploid cell. Meiosis 2 begins in the tertiary follicle and completed only if fertilization occurs. So this egg is kicked out in ovulation as a secondary oocyte, at which point meiosis 2 is going to be completed if fertilization occurs. But now look, we have a second polar body that is kicked out, and this will be degraded and broken down and not allowed to persist. This gives us 1 haploid ovum, a mature gamete that will occur. Again, if. So this is the mitigating circumstance, if fertilization occurs. So we can take our highlighter and let's highlight that red because this is the key factor if fertilization is going to occur. let's take a look at the histology of this ovary. So we can look at the ovarian cortex and we say, okay, here's our mesothelium lining the outside. We have our connective tissue sheath, the tunica albuginea. We have these little guys out here in the margin are our primordial follicles. The stroma, the cortex, is the tissue in between. And then we start seeing some growth. Notice it went a little bit bigger. There is still no antrum, no fluid-filled cavity, so we are a primary follicle. We're going to get bigger and bigger and bigger. We're going to form this zona pellucida, this purple layer around here. We are now entering into our secondary follicle. Look at the difference. We have a fluid-filled antrum. We have the formation of these granulosal cells surrounding the egg. We have our primary oocyte as the egg. Okay? And then we get to a Graafian follicle, which we'll look at one of our previous slides. But one more thing to identify since we have it here is the corpus luteum. So think about luteinizing luteum. Here we are pulsing hormones out, estrogen and progesterone. Okay, so these are our predominating hormones pulsed out. So what happens each month after that female hits puberty? So that gives us our two major hormones, like we saw in male reproductive, follicle-stimulating hormone and luteinizing hormone. This is what's going to stimulate the development of those primordial follicles. A few are going to start to grow. They develop into a primary follicle. However, one is only going to reach maturity. So every month when after puberty, so post-pubescent, every month, many follicles will be stimulated to grow under a surge of estrogen. So estrogen is going to cause those follicles, that's a signaling hormone, to cause those follicles to grow. But only one will be stimulated to continue to grow and grow. What happens to the other one? All those other eggs are destroyed. Let's take a closer look at a late primary follicle. We can see the primary oocyte. We have our formation of our granulosal cells. So again, these are our protecting cells. We need to protect that ovum. Again, remember, acidity. This is our enemy. The vagina is acidic, so we need to protect that egg from the vagina's ability to digest itself, or at least digest this egg. So those granulosal cells are going to be protective in nature. We have our basement membrane, and then as we become more superficial, we get our collagen fibers, our thecal folliculi, thecal cells, etc. Let's focus in on those granulosal cells. In the later stages of development, the primary oocyte is surrounded by several layers of granulosal cells. The glycoprotein zona pellucida forms between the primary oocyte and the granulosal cells. As maturation continues, the primary follicle develops into a secondary follicle. The thecal folliculi forms those stromal cells surrounding the outer portion of those granulosal cells. Now we look at a secondary follicle. Histologically, it looks different. We have the formation of the antrum, again, this fluid-filled cavity. We start seeing the zona pellucida enlarging. So we can see the zona pellucida enlarging, and the formation of the corona radiata, this radiating crown. Again, we have our thecal folliculi surrounding all our different cells. Now, a key difference in the secondary follicle is that the thecal differentiates into the theca interna and the theca externa. The innermost layer of granulosal cells become the corona radiata, this radiating protective crown. The secondary follicles become a mature Graafian follicle. This is the last stage, the largest stage of the follicle before ovulation as it pushes to the surface of the ovary. This is our mature tertiary or Graafian follicle. We have a nice fluid-filled antrum, right? So that antrum is filled with follicular fluid. We have that protective corona radiata, our primary oocyte. So we went from secondary oocyte to primary oocyte. We have our granulosal cells persisting. Remember, these are going to remain. These cells are going to remain to become that endocrine structure to pump out hormones. The theca externa and the theca interna. So interna and externa, again, setting up this follicle. To the right, we have a scanning electron micrograph of the primary oocyte, the thecal cells, and the granulosa cells that are forming that corona radiata surrounding it. Now we will continue with the rest of the female reproductive system in the second part of this lecture.